So, today I will talk in detail about action potential, and this is the very important question in your basic block physiology. So, in that first I will talk about the resting membrane potential, what is it, how it is uh, how it is established and maintained. Then I will enter into the then I will enter the action potential, and there in action potential I will show some definitions of the action potential with reference, and then I will talk about the uh, phases of the action potential, and then ionic basis of the action potential, and that then applied aspect also. In that you will be seeing that how it is transported in different nerve fiber, like in unmyelinated and myelinated nerve fibers, and then uh, a speed in different nerve fibers, that also I will talk in detail. So, hope you will go through the detailed lecture. Action potential. In action potential, let's first talk about the resting membrane potential. Sorry, resting membrane potential, RMP, and here I will in reference to talking about in reference to of neurons. Neurons, you know, resting membrane potential. So, what is it? At the term implies it is the potential at resting condition. Then resting condition, how will we define? Resting condition, and before entering moving in detail, I will ask some basic question to you. Let's see. Let's suppose it is question. Resting membrane RMP is the feature of RMP is the feature of your options will be nerve fiber or nerve cell, nerve fiber, B, muscle fiber, or muscle cells. Next will be virtually all cells, virtually all cells, and then option D is both A and B, both A and B. So, try to answer. What is your answer? Resting membrane is the feature of resting membrane potential is the feature of. Again, I will write next one more question before entering the answer. Action potential. Action potential is the feature or characteristic of again options will be same. nerve fiber, muscle fiber, virtually all cells and D both A and B. Options are same, but the question are different. Hope you have tried the answer and might have reached the right answer too. So let's review with your answer. Resting membrane pressure is a feature of the answer will be virtually all cells. This resting membrane, this resting membrane potential is across the all cell membrane, like not only the nerve fiber and muscle fiber or muscle nerve cell muscle cell. Obvious, these options are very correct, but some other cells like even in erythrocyte. Erythrocyte is a blood RBC and connective tissue. There also the resting membrane potential exists. That is potential gradient. Let's suppose this is cell. So here will be potential gradient, and this is the membrane. Here will be potential gradient cell. Potential gradient will be nearly in all cells, like your photoreceptors, rods and cones. There also, and there are important also there are nerve impulse transmitter from there also. But like in epithelial cells and other some other cells, uh, the resting event potential is the only of the local value. These cells will not transmit, and the structure these cells will not transmit the will not generate the action potential and not transmit the action potential unlike nerve fiber and muscle fiber, because there may not be the structure similar to the nerve fiber and muscle fiber, uh, that continuation that fiber like continuation, uh, voltage gated sodium and potassium channels and few other necessities. So the answer is virtually all cells. Then next question is action potential is a feature or characteristic of. So my dear, this question is very famous question. These two questions are very famous and basic question. If you have read the action potential, you must know these basic thing. These are basic thing you must know. The action potential is a feature of, then the answer correct answer will be both A and B. Only the action potential is and transmitted generated and transmitted in the nerve fiber and muscle fiber. So this is the this action potential is the feature of the nerve fiber and muscle fiber. Hope you understood. And again I would like to mention, while talking this, I will taking the reference 14th edition Guyton and Hall Essentials of Medical Physiology, and some other references are also. So main reference will be this Guyton and Hall. So you can refer this book, which edition is authorized in your territory. Now talk of about the resting membrane potential. So as the name suggests, it is the potential, it is the potential in unstimulated unstimulated or unstimulated or while not transmitting, while not transmitting the electrical signals, electrical signals. And you know better, the electrical signals in human body and other vertebrates are transmitted through are transmitted like electrical signals, electrical current. In same manner, the signals flow in nervous and muscular system of human body and other vertebrates. So, which is the factor that creates this resting end potential? Let us talk about that mechanism. Let us talk about the mechanism. Then first will be first point will be diffusion potential of potassium, diffusion potential of potassium across the membrane, across the membrane. And for this I will provide you a handwritten note also. So review that note also and the reference if you have any doubt. And further, if you have any doubt, in comment section you can comment your doubt. I will try to clear your doubt. Diffusion potential of potassium across the cell, across the membrane. So this is the diffusion potential of the potassium. How? Let's take an example. Let's suppose this is nerve fiber. Sorry. Let's suppose this is nerve fiber, and as you know, the membrane potential, the inside the membrane potential is negative and outside the membrane potential is positive. Rather than outside, we will talk about the inside potential, which is negative. Let's suppose this is nerve fiber, and here diffusion of potassium, will occur from inside to outside, inside to outside. Potassium. Actively, the potassium will be transported outside the cell membrane, and sodium will transported inside, sorry, potassium will transported inside the cell, and sodium will transported outside the cell by sodium potassium ATPase, sodium potassium pump. And diffusion occurs in either direction, diffusion occurs in either direction. So can occur in either direction, but mainly the diffusion occurs along the diffusion is the passive process and it mainly occurs along the concentration gradient or downhill movement. Downhill movement. So in that case, let's suppose all other channels, all only the potassium diffusion along the membrane or across the membrane is allowed and all other movement across the membrane is inhibited, is blocked. Then at that condition all other movements are blocked or stopped at some time. Then at that condition, you know, from basic physiology also, the potassium concentration inside the cell is higher and outside the cell is lesser. Let's suppose 140 milli equivalent per liter is here and outside it will be 4 milli equivalent per liter, and for same sodium here will be 14 milli equivalent per liter, and here in outside will be 142 milli equivalent per liter and this value, typically this sodium and potassium value outside and inside the cell or nerve fiber membrane is also asked in MCQ or Viva. So remember 4, 140, 14, 142, and sodium is lower molecular weight and potassium is a higher molecular weight. So molecular weight milli equivalency is potassium for is lower and sodium for higher. So reverse, that whose molecular weight is higher, their milli equivalent their concentration will be smaller, and in that sense also you can remember. So at that time only the sodium diffusion across the membrane is allowed, all other movements are blocked. Then diffusion of potassium occur, diffusion of potassium will occur till it creates minus 94 millivolt potential, inside the membrane. If in isolated condition, let us suppose only the diffusion of potassium is allowed and diffusion will occur along the concentration gradient, here is the higher concentration, here is the lower concentration, potassium will diffuse outside, that is in extracellular fluid, and this diffusion occurs till minus 90 millivolt potential is established inside the membrane, and this potential diffusion this potential which is the which is called diffusion potential of which is called diffusion diffusion potential which is called diffusion potential of potassium and this is the diffusion potential of the potassium. Now one more question here you can arise, that is the diffusion ceased once it reached at the equilibrium potential, once it reached at the equilibrium concentration or equilibrium, like this this concentration gradient is come to at equilibrium? No, the concentration gradient will still be there once this 90 minus 94 millivolt equivalent potential is established, but diffusion occurs before the equilibrium is achieved, before the equilibrium is achieved between inside and outside concentration of the potassium ions. Why? Why this occurs? Since diffusion is free movement and diffusion occurs until the equilibrium between two compartments, until the equilibrium is achieved, but here this will not happen. Why? Since this potassium ion is moving outside and outside, there will be excess positive charge, there will be excess positive charge because there are sodium and some other positive ions and this potassium, there will be excess positive charge. So this excess positive charge will repel, again will repel the diffusion of potassium, and potassium diffusion will be stopped because of this excess positive charge created outside the membrane. So here is not only the concentration barrier, there is charge barrier also, charge barrier also. This charge barrier inhibit at some point the diffusion of the potassium before the equilibrium is reached. So this is your knowledge point. If you are asked by anyone. So in that condition, how much potential will be established inside? Minus 94 millivolt equivalent, and this is the this again your will be your Viva and MCQ question, that diffusion of potential this mechanism this is the major or chief major contributor in establishing the RMP, resting membrane potential. Hence the RMP is more likely to be closure of diffusion potential of potassium, and in real life you can also be visualize this example, like in any field whoever contributes more, the right and um, governance is more towards him or her. So in that case, same case here you can see that measure more than 90 percent, more than 90% of the more than 90% part of the resting membrane potential will be established by this diffusion of the potassium alone, and there are diffusion of sodium also, and at few very less amount the contribution is also from the sodium potassium pump, and again the contribution of chloride ions should be also included, though it is in less amount, very less amount. So, this is the diffusion potential of the potassium across the membrane. Now, there are two more mechanisms that aid on this to establish the resting membrane potential. Resting membrane potential. So, then another factor will be number 2, another factor will be diffusion of sodium across the membrane that is due to diffusion potential of the sodium. Again, let's assume only the diffusion of sodium is allowed across the membrane. Then across the membrane here potassium, since is higher concentration in extracellular fluid, if all movement is blocked and the diffusion of the sodium is only allowed. So, diffusion of sodium will occur from outside to the inside, outside of the membrane to the inside along the concentration gradient, that is downhill movement. Then at this time sodium diffusion will establish or will create plus 61 millivolt potential inside the membrane, plus 61 millivolt potential inside the membrane if the diffusion of sodium is only allowed. And since this is positively charged ions, the diffusion of this sodium inside the membrane will cause the potential towards positive, and this value is frequently asked in the MCQ also. That diffusion potential of the sodium is how much. So, it will create plus 61 millivolt potential inside the membrane, Again, this diffusion is not stopped as the equilibrium is reached, but before reaching the equilibrium again same charge barrier will repel the diffusion of the sodium, and the diffusion potential diffusion potential stop somewhere plus 61 millivolt. Now, the tendency will be the membrane potential, the sodium will tend to create the membrane potential inside potential positive, but here is the mechanical barrier besides charge barrier, that this sodium leak channel this will occur through the leak channel. This sodium leak channel is very less permeable, nearly 100 times less permeable, 100 times less permeable than potassium ions, and the permeability of the sodium is very less in comparison to the potassium. It diffuse in very small quantity and the potential is not reached plus 61 milliequivalent practically 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 because the diffusion of the sodium is very less due to less permeability. So it will aid it will aid it will aid far less quantity in RMP, resting membrane potential, than the potassium diffusion. And there are one more mechanism you call it as sodium potassium pump. Sodium potassium pump. This sodium potassium pump this sodium potassium pump will aid on this sodium potassium pump or sodium potassium by this occurs by sodium potassium ATPase. So this pump is the feature of again all cells. In all cells this sodium potassium pump is present, and 30%, nearly 30% of the energy used by this pump by all pump which are present in our body. And this pump are very important functions and many important functions. I will talk in detail about sodium potassium pump. One feature is it establishes, it creates since it is electrogenic, since it is electrogenic pump, it creates nearly minus 4 milliequivalent potential inside the membrane of neuron, remember this is very less in comparison to potassium diffusion potential. How, why this is called electrogenic? Here 2 potassium and 3 sodium here will be ATPase action, and ATP will break down into ADP and inorganic phosphate. This ATPase activity will lead to the active transportation of the 2 potassium inside the membrane and 3 sodium outside the membrane, and each time you can see the 3 positive ions are pumped outside and only 2 positive ions are pumped inside. So there will be minus 1 negative charge deficit, negative charge deficit at each time. So this will also again aid to the establishing membrane potential and nearly equal to minus 4 milli equivalent, nearly 4 milli equivalent. So all these are all these are the factors that establishes the resting membrane potential, and this sodium potassium pump has more important in maintaining and re-establishing the resting membrane potential once it is disrupted by the action potential or or again subthreshold stimulus, or in other cells the stimulus causes the only change in the resting membrane potential, and action potential will not generate. Again the for maintain the outside and inside ion concentration of sodium and potassium, this pump will operate and re-establish or maintain the equilibrium, steady state. So the more role is re-establishing or maintaining the resting membrane potential by this sodium potassium pump, same as like that counter current multiplier and counter current exchanger you must have heard in renal block, and if you not, you will learn about this in renal block. Loop of Henle act as counter current multiplier system which establishes the hyperosmotic hyperosmotic interstitium, medullary interstitium, and that is maintained or re-established by the counter current exchanger mechanism that is called the counter current exchanger mechanism, that is Bhaja recta. I will talk in detail about that in renal and electrolyte block. So resting membrane potential, this resting membrane potential, till now we talked about the diffusion of the positive and negative charged ions, and by all these mechanisms this is created. But one thing, examiner might ask you that there is also some negatively charged particle inside the cell, like negatively charged proteins and some other ions, phosphates, sulfates, is the that also contribute to this resting membrane potential? The direct answer is no, but due to their presence inside the cells, here is occurring the net charge deficit. Since potassium is moving, positively charged potassium is moving outside the cell, the negative charged particles inside the cell, inside the membrane, will relatively get dominated. This negative charge is not by addition from inside, from outside the membrane, but there is a relative deficit of the positive charge, and the negative charge inside is in as it is quantity, there is no any addition, just the deficit of the positive ions, and that due to that deficit relative amount of the negative charge will increase, and that will create the negative potential inside the membrane. So direct role is of the diffusion of the positive ions outside the membrane, due to diffusion or this some amount by this sodium potassium pump, electrogenic pump. The direct, no there is direct any action, no the direct any action or role of this negatively charged particles inside the membrane or cell. And let's summarize this resting membrane potential. In summary, while operating, we talked to separate isolated diffusion of the sodium, potassium and sodium potassium ATPase. Now while all these mechanisms are operating simultaneously, it will create minus 90 milli equivalent uh, minus 90 millivolt potential inside the membrane, and among them 86% is caused by the potassium and sodium diffusion. Again, in that 86%, nearly almost all almost all contribution is from the potassium diffusion alone, and very less by the sodium diffusion, and nearly minus 4 equivalent by the sodium potassium pump sodium potassium active pump. And there are few roles of the chloride ions also chloride ions also. So while operating this mechanism, maximum potential can be created this much, minus 90 milli equivalent. And you know, the among all cells highest potential highest resting potential resting potential in quantity, not I am talking while not taking in sign, the minus 90 equivalent will be the of skeletal muscle. For example, here we can take some reference like for skeletal muscle skeletal muscle minus 85 to RMP will be minus 85 to minus 95, that is average 90 minus 90 milli equivalent. And this is the highest potential, and it will be as much as less in, let's suppose, in erythrocytes there is minus 10 milli equivalent 10 millivolt resting event potential. And other cells also varies, like in large nerve fibers the resting event potential is minus 70 milli equivalent. Large nerve nerve fiber, it will be again in range that minus 60 to minus 70 millivolt. And minus 70 millivolt will be of the large nerve fiber which is characteristic of this large nerve fiber resting event potential. And this is again your very important MCQ and Viva question, that typical nerve fiber or large nerve fiber has a resting event potential of minus 70 milli equivalent, sorry 7 minus 70 millivolt. And in the same way, like cardiac muscle, for cardiac muscle it will be minus 80 to minus 90 millivolt. For a smooth muscle, minus 50 to minus 60 millivolt. There are charts in the Gatton and Hall and other books also, you can refer that chart. And this potential sometimes will be asked in MCQ and Viva. So this is all about resting membrane potential. Video got longer because I want to clear the concept, so more time is consumed in that. I am sorry for consuming your more time, but once your concept is cleared, once your concept is clear, if you clear one concept in 1 hour clearly with crystal clear concept, then you have not look for the many videos or many lectures, many references, many books. So remember, the target of FA medical lecture will be clearing the concept, and once you are clear each concept, you can answer SAQs, MCQs, Viva all. Now move on to the action potential proper.